Hi, and welcome to the WellnessFX trading video, where we talk about some cardiovascular and inflammatory risk markers. Specifically, we'll be looking at free fatty acids, lipoprotein-associated phospholipase A2, apolipoprotein A, homocysteine, and fibrinogen. Let's first start with free fatty acids. Now, to really talk about what a free fatty acid is in the first place, you have to understand that in our body we have things called triacylglycerols or triglycerides. A triacylglycerol is a glycerol molecule bound to 3 fatty acids. And all a fatty acid is just a series of carbon chains, a certain amount of, you've heard of saturated or unsaturated fatty acids, for example, and that's really what a fatty acid is. So a triglyceride or triacylglycerol are 3 fatty acids bound to a glycerol molecule. Now those fatty acids, if not bound to a triglyceride or triacylglycerol, because they're free, are referred to as free fatty acids. Now, the majority of the triglycerides in our body are stored in an adipocyte, or fat cell. Inside of the fat cell, we have a number of these triacylglycerols, or triglycerides, which again is really just a glycerol molecule and a fatty acid. If something were to break one of these fatty acids off of the glycerol molecule, it could go into the bloodstream and would be called a free fatty acid. And that's what this particular marker is looking at, is serum or blood levels of free fatty acids. So let's talk about what this really indicates and physiologically what may be going on in somebody that has high free fatty acids. There's really 2 things that may cause high free fatty acids in the blood. Within adipocytes, there's an enzyme. The enzyme is called hormone-sensitive lipase. And as the name would imply, it's sensitive to certain types of hormones that, when those hormones are present, will go to the triacylglycerols and actually break off some of these fatty acids, making free fatty acids and delivering them into the bloodstream. Hormones that will do this, for example, are epinephrine and glucagon. Now, glucagon is a hormone that's made by the pancreas in response to low glucose. Just think about it, if glucose is gone, the pancreas makes glucagon in order to help increase glucose levels. If glucose is low and glucagon is high, that's indicating sort of an energy deficient state. If glucose is low, there's not an abundance of it, and we should actually break down tissues in order to help fuel the body to make more glucose, for example. Conversely, if glucose is ever high, the pancreas makes the hormone insulin in order to help store things. And so in the presence of high glucose, that's an energy-rich state, and we don't want to break things down in the body, rather we want to help store them. So as it pertains to hormone-sensitive lipase, if insulin is ever binding on and attaching to the receptors found on fat cells, if insulin is present, that generally means that glucose is high, and therefore hormone-sensitive lipase will stay put. There's no sense in breaking down fatty acids and triglycerides within a fat cell if there's an abundance of energy as indicated by excess insulin. So again, if this fat cell has a receptor for insulin and insulin binds onto this, then that's signaling to the cell there's an abundance of energy right now. Hormone-sensitive lipase should stay put, stay in place. Don't break anything down, because we're right now in a period of storage. Conversely, if the receptor of the cell, the fat cell, receives glucagon, then the message that's being sent to the fat cell is there's an energy deficient state right now. Hormone-sensitive lipase can then translocate or move to the triglycerides, break down these fatty acids, liberate them into the body, which is a good thing because we are in an energy deficient state and therefore need to break things down to help maintain the energy levels. So here's what happens. In the case of insulin resistance, despite the fact that somebody may have an abundance of glucose, if over a period of time their insulin stops working because the insulin receptor sites start to downregulate, causing insulin resistance, even though somebody may have high levels of insulin, high levels of glucose, because insulin is not accurately signaling this cell because the receptor doesn't work, inside the cell, hormone-sensitive lipase does not receive that signal that insulin is present and we should be storing things, and instead moves to help break down these free fatty acids. Now, these free fatty acids have been shown to increase one's risk for things like insulin resistance, metabolic syndrome, type 2 diabetes, as well as cardiovascular disease. And there's a number of different mechanisms as to why. One of them is considered to be lipotoxicity of the pancreas, for example, that the beta cells, which normally make insulin, start to accumulate excess fats, free fatty acids, because it was being liberated out of the cells, and causes lipotoxicity and then issues with the pancreas. And there's also some evidence that free fatty acids actually contributes to even more insulin resistance, metabolic syndrome, type 2 diabetes, and of course, with those that are present, then cardiovascular disease. So elevated free fatty acids can indicate that there is insulin resistance taking place because insulin is not working. It's not telling the fat cell to keep hormone-sensitive lipase in place, and instead then pretends that there is a deficiency of energy, starts to break down the free fatty acids, have them be liberated from the fat cell and into our bloodstream. So high free fatty acids is thought to be a bad thing as it pertains to insulin resistance and some of those things. But here's the other thing. If somebody is following a very low carbohydrate diet, almost a ketogenic diet, what they have then is they'll have low glucose and therefore high glucagon. And if they have low glucose, they'll also have low insulin. And if you go back to this model here, low insulin and high glucagon stimulates hormone-sensitive lipase to go to these triacylglycerol molecules and break down and release free fatty acids. So you can really see elevations in free fatty acids in the bloodstream of two individuals, and it's a very different story. One may be in an insulin-resistant individual. Another one might be in somebody that's following a very low carbohydrate diet, has fairly low, and maybe even a ketogenic diet, low glucose levels, low insulin therefore, and high glucagon. Now, how do you pick this up? In the individual with insulin resistance, they may have high free fatty acids, but then also have high glucose, high hemoglobin A1C, as well as high triglycerides, especially in the triglyceride to cholesterol ratio. In an individual that's following a low carbohydrate or ketogenic diet, they will likely have relatively low glucose, serum glucose, possibly low hemoglobin A1C, depending on how long they've been following this diet, and possibly low triglycerides as well. So are free fatty acids in the bloodstream good or bad? The research would suggest that it's bad, and that it actually contributes to things like insulin resistance. However, in an individual that's following a low carbohydrate, low calorie diet, they will likely be breaking down fat cells for use of energy for the cells of the body, which is a good thing. Does this potentially cause insulin resistance in those individuals? It hasn't fully been elucidated. So, elevations in free fatty acids on a blood chemistry, you may be looking at insulin resistance, and you can pick that up by seeing high free fatty acid levels as well as high glucose, high hemoglobin A1C, possibly high triglycerides, especially triglyceride to cholesterol ratio. In somebody else, where they're actually breaking down their fatty acids and using them for energy, you may see low glucose, low or normal hemoglobin A1C, low triglycerides, although you may find free fatty acids elevated on a blood chemistry. So now let's look at lipoprotein-associated phospholipase A2. Now let's talk about lipoprotein-associated phospholipase A2, abbreviated LPPLA2. Lipoprotein phospholipase A2 is a relatively newer marker when looking at cardiovascular disease risk, and it's a very interesting marker, and there's actually some controversy surrounding it. But the evidence really does point to that if the lipoprotein phospholipase A2 is elevated on a blood chemistry, that somebody is at risk or increased risk for cardiovascular disease. And here are some reasons why. So within the blood cells, we have low-density lipoprotein particles. These particles contain phospholipids, triglycerides, some cholesterol, the apolipoprotein B, for example. And this is a particle that's floating around containing all this stuff. So these are LDL particles, low-density lipoprotein particles. Low-density lipoprotein particles contain this particular enzyme called lipoprotein phospholipase A2. It's thought that this particular enzyme is manufactured by immune cells like macrophages and some T cells, for example, dendritic cells as well. That this enzyme manufactured by our immune system cells is incorporated in these low-density lipoprotein particles. Now, here's where the controversy exists. Lipoprotein phospholipase A2 has been shown to help improve oxidized LDL cholesterol. In other words, the oxidized LDL cholesterol is shown to be atherogenic and inflammatory. And lipoprotein phospholipase A2 has been shown to actually help improve the oxidized LDL. In other words, to hydrolyze or remove some of the oxidized LDL, which is a good thing. What happens, however, is in the process, other inflammatory cascades are taking place. For example, platelet activation, platelet aggregation, and therefore sort of an inflammatory response. Now also, within the tissue, the vascular walls, we have things like macrophages, which, as we talked about in other videos, macrophages will actually start out as monocytes, go into the tissues, become macrophages, start to consume or phagocytize some of the oxidized LDL and components found within the vessel wall. And they too can produce lipoprotein phospholipase A2 that actually helps increase smooth muscle aggregation to this area, which is seen very commonly in the atherosclerosis process. So, lipoprotein phospholipase A2 is therefore seen as somewhat of an active indicator of endothelial dysfunction. How much endothelial dysfunction is actively taking place? And the reason why is because this particular enzyme will be found in higher amounts if the macrophages and T cells and dendritic cells are producing more of it. The question is why are they producing more of it? Chances are because there's inflammatory processes that are occurring around the cardiovascular system. So if lipoprotein phospholipase A2 is elevated, it's thought that yes, there's an increased risk for cardiovascular disease, but it's a marker of sort of active endothelial dysfunction and therefore active atherosclerosis or atherogenic processes within the vascular walls. In terms of if it's good or is it bad, the question of if it's good or if it's bad is when you look at lipoprotein phospholipase A2, again, why is it produced in such high amounts? Because there's probably inflammation as well as high amounts of oxidized LDL. So in somebody that has higher oxidized LDL, perhaps the body is trying to protect itself from that oxidized LDL, making or manufacturing more of this particular enzyme to help deal with that oxidized LDL, but unfortunately in the process is producing other inflammatory processes that increases one's risk of cardiovascular disease. So the bottom line is, it's a relatively newer marker when looking at cardiovascular disease. The research thus far is pretty clear that if lipoprotein phospholipase A2 is elevated, that somebody is at an increased risk for cardiovascular disease. And it's thought that because it's looking at active endothelial dysfunction and the inflammatory process that's contributing to atherosclerosis. So now let's look at apolipoprotein A. Now let's talk about apolipoprotein A. And if you watched the video where we talked about apolipoprotein B, apolipoprotein A is essentially a similar conversation. That what we have here is an HDL particle, not HDL cholesterol, but an HDL particle. As we've discussed with these particles, what a particle really is a molecule largely synthesized by the liver and some other tissues that is surrounded by phospholipids. And the reason why is this lipoprotein particle is a transporter for things like fatty acids, triglycerides, cholesterols, and some other things. To be able to be transported in the otherwise aqueous solution of our blood or plasma, it's coated with phospholipids. Phospholipids can exist very well in an aqueous environment. Fats, on the other hand, cannot. So the HDL, the high-density lipoproteins, which are considered to be the good lipoproteins or good cholesterol, are the smallest of the lipoproteins. And it's considered to be good because these will actually pick up cholesterol and pick up fatty acids en route back to the liver to be metabolized and broken down. Now, associated with some of the HDL particles, or I should say, now associated with HDL particles, but not exclusively HDL particles, is a protein called apolipoprotein A, or sometimes apolipoprotein A1. Now, if you remember the conversation about apolipoprotein B is considered to be a proxy or surrogate marker for LDL particle numbers, and that's because every LDL particle has an apolipoprotein B molecule or protein associated with it. Now, that's not true of the HDL or high-density lipoprotein particles. That the apolipoprotein A or apolipoprotein A1 is associated with HDL particles, but it's also found on other ones. So it's not a one-to-one surrogate or proxy marker of HDL particle numbers. However, because it is associated with HDL particles, that it is considered to be a marker looking at the relative abundance of the HDL particles. So if the apolipoprotein B levels are elevated, then that indicates that there's likely a high amount of LDL particles. If the apolipoprotein A1 is elevated, then that indicates that there's likely but not as exclusively elevations in the HDL particles, which would be good. However, again, it's not a one-to-one exclusive ratio because this same protein, apolipoprotein A or apolipoprotein A1, is found on a variety of different types of particles, HDL being one of them. So to be sure, to really evaluate the apolipoprotein proteins or molecules, if the apolipoprotein A to the apolipoprotein A1 ratio is really one of the best indicators in terms of that model for cardiovascular risk. The greater the ratio, meaning the higher the apolipoprotein B to the apolipoprotein A1, the greater the one's cardiovascular risk because there's more LDL particle numbers and less HDL particle numbers. The closer the ratio, in other words, the lower the apolipoprotein B, the higher the apolipoprotein A1, shows that there's a relative lowering or decrease of the low density lipoprotein particles and an increase in a number of particles, including the HDL particles, thus reducing one's cardiovascular risk. And according to research, the ApoB to ApoA1 ratio seems to be a really good indicator in terms of one's cardiovascular risk. So that's the short story on the apolipoprotein A1. The higher the apolipoprotein A1, it's generally thought the lower one's cardiovascular risk because of its association with HDL particles, and therefore the higher the apolipoprotein A1, the higher the HDL particle count. So now let's talk about homocysteine. Now homocysteine is a very interesting compound. It's been the result of a lot of different controversy over the past couple decades or so, because on the one hand, it's been fairly well established that elevated homocysteine, which is a protein amino acid, has been associated with increased risk for cardiovascular disease, endothelial dysfunction, certain neurodegeneration processes, and so that's pretty well established. But at the same time, and I'll show this pathway in just a second, it's been shown to be very easily managed or lowered in its numbers on a blood chemistry by some fairly basic B vitamins. But what hasn't been shown is simply lowering homocysteine, which has been shown to increase one's risk for cardiovascular disease, doesn't necessarily decrease one's risk for cardiovascular disease or cardiovascular mortality. So the jury is still out about homocysteine. In my mind, it is a damaging molecule. It's an indication of a dysfunctional process, and what's the methionine sort of cycle, which is a normal amino acid to have in the body. And to try to lower it is probably a good thing. However, by itself, if simply lowering it, probably is not going to take care of all disease risk, and really, which you know that we really should be taking a look at just about all the different processes of the body for trying to reduce disease and optimize health. So essentially, here's the quick story on homocysteine. Homocysteine by itself seems to be a damaging molecule. Like I said, it has been shown to damage vascular endothelium, causes neurodegeneration in some elements of the brain, but homocysteine can fairly easily turn into the amino acid methionine. And methionine is obviously, as an amino acid, far less damaging than homocysteine. To do so requires the active form of vitamin B12 called methylcobalamin. And what we're really showing in this pathway here is essentially the transfer of methyl groups. And methylcobalamin, if you notice, homocysteine doesn't have a methyl group. But as the name methionine would suggest of this amino acid, that it does have a methyl group. It gets this methyl group from methylcobalamin. Methylcobalamin, under the influence of an enzyme, will donate its methyl group to homocysteine. Now homocysteine has the methyl group and it turns into methionine. And now active form of B12, or methylcobalamin, turns back into cobalamin. If this only happens by itself and cobalamin cannot be regenerated into methylcobalamin, you can see how homocysteine levels would increase. So a B12 deficiency can cause an increase in homocysteine because if you lack active vitamin B, or specifically methylcobalamin, you can't convert homocysteine into methionine. The other story, though, is how does inactive cobalamin, or B12, turn back into active methylcobalamin? Well, all you need to do, if you notice the names again, is add a methyl group to cobalamin. And a really great way of doing that is B12's friend, folic acid. Now the active, or one of the active forms of folic acid in the body is called 5-methyl tetrahydrofolate. So you can see here that this particular form of folic acid has a methyl group to donate. And similarly, 5-methyl tetrahydrofolate donates its methyl group to cobalamin, regenerating cobalamin, or B12, into active B12 called methylcobalamin, and in the process, 5-methyl tetrahydrofolate turns into tetrahydrofolate without the methyl group because it was donated to reactivate cobalamin. So you can see how high homocysteine can be because of a B12 deficiency. Without B12, you don't have methylcobalamin to donate the methyl group to homocysteine to turn into methionine. Conversely, you can also have high homocysteine due to folic acid deficiency, because without folic acid deficiencies, you may have B12, but you can't actively methylate it, and therefore you'll have low levels of methylcobalamin, low ability to methylate homocysteine to turn it into methionine. So you can see that either or B12 or folic acid deficiency can contribute to high levels of homocysteine, and indeed both of those have been shown to decrease homocysteine levels. The other pathway here that's noteworthy is in the presence, it's a couple of steps, but in the presence of B6, vitamin B6, homocysteine can go down a different pathway to turn into another amino acid called cysteine. Now, cysteine can turn into, later, taurine, or perhaps more importantly, glutathione. Now, you can see that if homocysteine is elevated, it's really a possibility of three reasons. One is because lack of active B12. Two is because of a lack of folic acid. Or three, in this alternative pathway, it could be a lack of vitamin B6. And again, according to research, all three of those vitamins have been shown to lower homocysteine levels. Now, does that confer cardiovascular benefit or health? And again, the controversy still remains, and that hasn't been answered. But in the meantime, knowing that homocysteine causes damage, that if it's elevated on somebody's blood chemistry, you might be looking at either a B12 or folic acid or B6 or all of the above or some combination deficiency. And simply making sure that they have an adequate, usually B vitamin that contains at least these three can help lower homocysteine. In the course of 3 months, you can retest and see if homocysteine has been lowered. Last marker we'll look at in this particular video is fibrinogen. So now let's talk about the last marker in the inflammatory panel, and that is fibrinogen. Now, first of all, anytime a protein or enzyme ends in inogen, it usually refers to the inactive form of that protein or that enzyme. So fibrinogen is a plasma protein found in the plasma that's synthesized in the liver, along with a number of other proteins. However, fibrinogen is about 4% of the total plasma proteins, which is actually quite a bit when you consider how many proteins are actually found in the plasma. So fibrinogen is an inactive, soluble plasma protein made in the liver that's circulating ideally in all of our bodies right now. And we use fibrinogen anytime there is damage to the vascular endothelial layer. If there's maybe a microvascular rupture in a capillary, or perhaps it's in a larger artery or blood vessel, then fibrinogen is going to be used. And here's why. As was talked about in another video, we have platelets that are generally inactive circulating around the body, and anytime there is damage or exposure to the basement membrane, these platelets become active and go in order basically become sticky and stick together in the site of damage. And this is the initial plug called the primary platelet plug that blocks any excess blood flow from or blood cells from leaving in this particular area of damage. Now, this is great. However, it's only temporary. Because this is called a primary platelet plug, we need to figure out a way that we can actually really keep these platelets in place to form a more permanent plug, if you will, so that this endothelial tissue can actually repair itself back to normal. And that thing that's going to actually really keep these platelets in place is called fibrin. Now, there's this fairly complex cascade in the body called the clotting cascade. And essentially what it is a number of inactive clotting factors, all proteins made in the liver, that in the presence of damage and some activating substances become active. And all of this cascade, for example, clotting factor 12 activates clotting factor 11, which activates clotting factor 9, and et cetera, et cetera. But you have to see that whole story, because down here, clotting factor 2 is called prothrombin, becomes activated into thrombin, and thrombin, as an active protein now, activates fibrinogen, which was a soluble, inactive protein found in the plasma, and turns it into the active, insoluble form called fibrin. And what fibrin does now is it forms this mesh network over this primary platelet plug to create what's called a secondary platelet plug. That this insoluble fibrin protein sort of stitches this area up, forms this mesh network, so now these platelets aren't going anywhere, contracts itself tighter, allowing blood flow or blood loss to really stop, as well as the endothelial layer to actually repair itself. So, the marker, however, on a blood chemistry is fibrinogen. Now, as you can see, because fibrinogen, when active, turns into fibrin, and fibrin is used in the clotting process, that an increased level of fibrinogen is an increased risk for cardiovascular disease, because more fibrinogen means potentially more fibrin, which potentially means more clotting, and excessive clotting can lead to occlusion of blood vessels, or it can lead to something like a thrombus or embolus, where this breaks free and ends up occluding a vessel somewhere else, causing a heart attack or a stroke. So the short version is increased levels of fibrinogen increase one's risk for cardiovascular disease because of the increased clotting that's potentially taking place that can lead to things like cardio or heart disease, heart attacks, or stroke. So, if somebody has an increase in fibrinogen, why might it be high? Well, there's a number of different proteins synthesized in the liver that are considered to be acute phase reactants. For example, you may have heard of CRP, talked about in another video, or ferritin, which is talked about in a different video. Fibrinogen is another acute phase reactant, meaning that in the presence of inflammation is produced at a higher than normal level or higher than normal amount. So if fibrinogen is high, you may really just be looking at some kind of inflammatory response that's occurring in the body. You'd know it was an inflammatory response by looking at things like ferritin as well as CRP, and if those are both high, then it's likely elevated due to some kind of active inflammatory response. Now, on the other hand, which is interesting to consider, if fibrinogen levels are ever low, now it's probably not much of a consequence in terms of overall health and cardiovascular risk. However, it can be a risk for increase in bleeding. And if fibrinogen levels are excessively low, you'll probably want to refer that individual out to their doctor to take a better look and do some follow-up lab testing. But reasons for high fibrinogen will typically be some kind of inflammatory process, sometimes even malignancy. Never say that to a client, but just understand that that's usually the reason why that they'll be elevated. One other thing to consider, however, is it is possible to theoretically have normal levels of fibrinogen because, even though an abundance is being made, but because it's all actually being used up, turning into fibrin. The more fibrinogen that turns into fibrin, the lower levels of fibrinogen will actually show on a blood chemistry. So just consider that even though fibrinogen may be normal, if there are other inflammatory markers that are high, consider that fibrinogen may look normal or maybe even low because it's actually being all used up and activated into fibrin, and the body hasn't been able to compensate in terms of making more fibrinogen. So high fibrinogen definitely increases one's risk for cardiovascular disease, but normal fibrinogen doesn't mean they're not at risk, and you really have to look at the rest of the blood chemistry to see if there's other cardiovascular risks, in which case you might be able to make the assumption that if things like lipoprotein phospholipase A2 is high and there's active endothelial dysfunction, that maybe fibrinogen is normal because it's being converted over to fibrin so actively and so quickly. So that wraps up the markers on this particular video, and I will see you in another video.